This next recording will be going over blood grouping and evaluation of laboratory or blood work. So, blood grouping refers to identifying antigenic molecules which are found on the surface of red blood cells. Red blood cells have on their surface a series of carbohydrate molecules, complex carbohydrates, which are antigenic, which means that the immune system recognizes them and would make antibodies against them. Turns out these molecules can come on one of two versions. Some cells have a complex carbohydrate of the type that we have come to call A on their surface. Other red blood cells have a different carbohydrate, and these are complex carbohydrates. In this case, we have come to call this second form of carbohydrate B. Other individuals carry in their blood a combination of both, the A complex carbohydrate and the B complex carbohydrate. We say that these individuals carry the blood type AB. Yet there are other group of people who carry neither of these carbohydrates on their surface. We call these group O. Now, the importance of these complex carbohydrates, which again are antigenic and stimulate the immune system, is that these molecules are not only found in blood, but they're also found on the surface of other types of cells, like bacterial cells or fungal cells, or even some type of plant cells, food. What that means is that the minute that we're born, we begin to be exposed to these bacterial and fungal cells, and our body may be exposed or will be exposed to a foreign complex carbohydrate, different from the one we carry on our blood cells. So, an individual who has the A complex carbohydrate on the surface of their red blood cells, when exposed to the B carbohydrate, their immune system will begin to make antibodies against the B carbohydrate. An individual who's carrying the B carbohydrate on the surface of the red blood cells, when exposed to the A carbohydrate through bacteria and foods, their immune system will respond by making the anti-A carbohydrate. The anti-A antibody. These antibodies will be circulating in the plasma of the individual. A person who has both A and B carbohydrates, because they are carrying both types of antigens, their immune system will not make antibodies against any of them. On the other hand, those individuals who carry neither of these carbohydrates will, when exposed to A and exposed to B carbohydrates, will make antibodies against both. So they will carry anti-A and anti-B antibodies in the blood cells. So individuals that are typed as having blood type A have the anti-B antibody circulating in their plasma. Individuals whose blood type in the red blood cell is B, in their plasma they're going to have anti-A circulating. Those who carry both A and B antigens in the red blood cells and have the type AB will carry neither antibody in the blood cell, in the blood plasma. Those who are type O will carry both the anti-A and anti-B. Now, antibodies carried in the plasma, if they encounter the antigen that they can bind in a red blood cell, they will bind to the antigen, they will clump the red blood cells and target them for destruction. That is why it's crucial when an individual, a patient, receives a blood transfusion, the blood needs to be matched to a compatible blood group. So individuals that are of the blood group A will be able to be matched to type A blood. So this is match. Type A's can match to the A type blood. If given the type B blood, the anti-B circulating in their plasma will destroy the B cells, so they could not be given type B blood. Type AB blood also has the B antigen, therefore the anti-B circulating in the type A blood will destroy those cells. However, the O type doesn't have any of the antigens, therefore the anti-B circulating in the plasma of the recipient will not affect the new blood received. So a person with type A blood could receive type A or type O blood. Similarly, a patient with type B blood can be matched with someone with type B or someone with type O. A patient with type AB could be matched with someone with type A, someone with type B, someone with type AB, or someone with type O. And an individual with an O type, because they carry the anti-A and anti-B antibodies in their plasma, could only be matched with type O's. Notice that type O is a match for everybody. That's why type O is called the universal recipient. On the other hand, type O is the universal donor. Type O blood is the universal donor. On the other hand, type AB can only be given to another AB. But a person with type AB could receive any type of blood. So that is called the universal recipient. So type O is the universal donor. It can donate to anybody. Type AB can receive any blood. When blood is transfused, the only part of the blood that is transfused to the patient are the red blood cells, not the plasma of the patient. So we don't have to worry about the plasma of the donor, because the only thing that is transfused is the red blood cells of the donor, not the plasma. So from here you should know that type A blood carries the type A antigen and the anti-B antibody. Type B blood carries the B antigen and anti-B antibody. Type AB blood carries the AB antigen and non-A or the B antibodies. And type O blood carry none of the antigens, therefore will make anti-A and anti-B antibodies. So keep in mind the importance of the ABO system is that these antigens are common in the living world. They're what we call heterogeneous antigens. because they're present not only on human red blood cells, but in many other types of cells. So even if we are never transfused any type of foreign blood, our body will still be exposed to these antigens via bacteria and fungus and other cells. So this gives you again a depiction of what we have talked about. This is the antigen and this is the antibody carried by type A blood. Antigen and antibody carried by type B blood. Antigens and no antibodies carried by type AB blood. And then again, no antigens in the case of O, which means they carry both the A and B antibodies. So when we cross-match blood for transfusion, we're going to mix the serum of the patient, the plasma of the patient, with the red blood cells of the donor to make sure that there are no antibodies in the recipient that could potentially create a problem. So this is a type A donor, not donor, sorry. I guess that is type A blood. Okay, so a recipient who is type A, the plasma of the recipient is going to be matched with the red blood cells of the donor. If the plasma of the recipient has no antibodies that can bind to the donor's red blood cells, then we have a good match. On the other hand, if the recipient, in this case is type B, because it carries a type A antibody, if the plasma of the recipient, when mixed with the red blood cells of the donor, the recipient has antibodies that match and bind to the donor red blood cells, that is considered an incompatible match, because the same binding will occur inside the body of the recipient, and it will cause destruction of the donor red blood cells. Okay. So the donor is the person who donates blood. The recipient is the person who receives the blood. Blood transfusions do not involve all of the blood, but only part of the blood. So what is transfused are the packed red blood cells, not the plasma of the donor, just the red blood cells of the donor. Blood is then fractionated, and the red blood cells are separated from the white blood cells and from the plasma and from the platelets, and even from some of the clotting factors. So all of the fractions of the blood can be used and given to different patients that may need them. Okay, so we have talked about what we call the ABO system. The importance of the ABO system is that these molecules, again, are common in the living world. You may have also heard of the RH system. The RH system is very different. We're talking now about a protein that is found in red blood cells, only in red blood cells. We do not find this protein in any other type of cell except that of human cells, and actually also a type of monkey called rhesus monkeys. And that's why the RH system, this type of protein system that is found in red blood cells, is called RH because of rhesus monkeys. The word rhesus starts with RH. So this type of protein in red blood cells of humans and rhesus monkeys are again unique to these red blood cells. So the protein itself is called the A protein. Sorry, not the A, the D protein. Let me go ahead and go to the previous one really quick. So the protein itself is called the D protein. Okay. So the antigen is called the D antigen or the D protein. The blood group is called the RH group, but the protein that is the actual antigen in the blood group is called the D protein or the antigen D. Notice that a large percentage of the population carries this protein in their red blood cells, 85 to 88%. So most of the population will have the D protein and they will be classified as Rh positive. A person who lacks antigen D are called Rh negative, and these individuals will make an antibody against the D protein if exposed to the antigen. And this is a very important concept because the only time the antigen is made is if the blood cells carrying the D antigen are present, are exposed. Otherwise, an Rh negative individual will never make an anti-D antibody. So antibodies develop only after exposure to the D antigen. So Rh positive individuals already have the D antigen, they're not going to make the antibody. Rh negative individuals, because they don't have the antigen, if exposed to blood that is Rh positive, they will make an anti-D antibody. For this reason, Rh positive blood is never transfused to Rh negative patients. So when we match blood, we not only match by the ABO system, but we also match by the Rh system. So an A negative patient can only receive A negative and O negative blood. An B negative patient can only receive B negative and O negative blood. An O negative patient can only receive O negative blood. An AB negative patient can only receive A negative, B negative, AB negative, and O negative blood. Whereas an A positive patient could receive an A positive patient can receive A positive, O positive, A negative, and O negative. So an O positive patient can receive Rh positive or Rh negative blood. Same for a B positive patient can receive B positive or O positive, but he can also receive B negative or O negative. An O negative patient can only receive O negative blood. An AB negative patient can receive A negative, B negative, AB negative, or O negative. However, an AB positive patient can receive any type of blood. These are called the universal recipients. AB positive is the universal recipient. It can receive any type of blood, Rh positive or Rh negative. A O positive blood can receive O positive or O negative blood only. So because O negative blood can be given to anybody, this is called the universal donor. O negative is the universal donor, AB positive is the universal recipient. Now, the Rh system has a special implication in pregnancy. An Rh negative mother carrying an Rh positive fetus may develop the anti-D antibody, which could harm the Rh positive baby in the uterus. And this is the special consideration that Rh negative women needs to have upon pregnancy. So these Rh positive babies being carried by an Rh negative mother may develop a condition called hemolytic disease of the newborn. What happens is that during pregnancy, especially towards the end of the pregnancy, small hemorrhages exposes the mother to the D antigen, the Rh positive fetal blood. The immune system of the Rh negative mother then begins to make anti-D antibody. Because these type of antibodies can cross the placenta, the anti-D antibody can go into the fetus's blood system and begin destroying the fetal red blood cells. Because this typically happens towards the end of pregnancy, the babies are born severely anemic, severely jaundiced. However, part of the problem is that once the mother has begun making anti-D antibody, she will continue to do so pretty much throughout her entire life. If a second pregnancy ensues with a baby that is Rh positive, from the start of the pregnancy, the antibody of the mother will start to cross the placenta and destroy the baby's, the fetus or the embryo's red blood cells. So the second pregnancies are particularly a danger of a spontaneous abortion or a pregnancy that will not be carried to term. So you should know what it is, hemolytic disease of the newborn. And this slide goes into what is happening. These green Y shaped structures are the antibodies of an Rh negative mother, which can cross the placenta, and they are here seen in the body of the fetus, destroying the fetal red blood cells. And again, the first pregnancy is typically okay with a little bit of problems, a little bit of anemia or jaundice. It is the second pregnancy that becomes very, very risky at this point because the mother continues to be sensitized. Now, there is something that mother can do. There is a medicine called RhoGAM, which is used to prevent hemolytic disease of the newborn. Turns out RhoGAM is actually the anti-D antibodies, exactly the antibody we don't want the mother to make. That's what we're giving the mother early in the pregnancy or after delivery of the first baby. The medicine is called RhoGAM. And again, it contains exactly the antibodies we don't want the mother to make. Okay, let me show you why RhoGAM works. So if there are Rh positive blood cells from the fetus being released into the mother's bloodstream, and the mother's being given RhoGAM, RhoGAM is going to bind and hide the Rh positive and D protein in the fetal cells. So that because now they are covered with antibodies from the RhoGAM, the immune system of the mother cannot be exposed to the D protein, and she will not make her own antibodies. So we're tricking the immune system by hiding the antigen with the RhoGAM antibodies so that the mother's immune system does not see the antigen and it will not make her own antibodies against the D antigen. So that's how RhoGAM works. Okay, so you should know what RhoGAM is and what it contains. So why would a B positive blood can be transfused into a person with A negative blood? Because a B positive person will have anti-A antibody in their plasma, and they will destroy the A antigen of the recipient. Which ABO-Rh blood type will be considered universal blood donor. Universal blood donor will be the O negative. The universal blood recipient is the AB positive. And you should know what is hemolytic disease of the newborn, which is the condition that arises from an Rh negative mother carrying an Rh positive fetus. And RhoGAM is the anti-D antibody given to the mother to prevent her immune system from developing her own anti-D antibodies. Okay, and the final portion of this chapter looks at blood evaluation and abnormal conditions. Now, be familiar with or have an idea of the numbers of red blood cells in circulation per macroliter. So we're talking about millions of red blood cells, 4.6 to 6.2 in males, 4.2 to 5.4 in females. So you should know that 2 million red blood cells per macroliter is way too low, and 60 million is way too high. So you should have a rough idea of the number of millions of red blood cells per macroliter of blood. Hemoglobin measurements, again, just have a rough idea, 14 to 18 grams per 100 milliliters in males, 12 to 16 grams per 100 milliliters in females. So you should know that 10 is too low, 20 is too high. Now, the hematocrit is a very simple and inexpensive test that can be done very cheaply at a doctor's office. It consists on drawing blood into a very thin capillary tube. Then the blood is spun at high speeds to separate, so that the red blood cells, will be separated from the plasma. Under normal conditions, the percentage of plasma in the blood will be about 55% of the total. The percentage of red blood cells will be about 45% of the total, give or take. Hematocrit is the measure of the percentage of red blood cells when the capillary tube is spun. So in this example, the hematocrit is 45. Males can have a hematocrit of 40 to 54%, whereas females have a slightly less hematocrit, 38 to 47. So a hematocrit of 40% red blood cells means there's 60% of plasma, and that's a normal range. A high hematocrit, meaning more blood cells than plasma, typically occurs when there is dehydration, because it is not a matter of more red blood cells, but just less fluid, which gives a higher percentage of the red blood cells. Low hematocrit typically means anemia or a low red blood cell numbers due to perhaps something like bleeding. Okay. So you should know what is a hematocrit and have an idea of what are the normal hematocrit levels and what does a high hematocrit versus a low hematocrit represent. White blood cells are counted in thousands per macroliter, 5 to 10,000 white blood cells per macroliter. A condition of low white blood cell is called leukopenia. This may be due to bone marrow suppression, viral infections, or vitamin B12 or folate deficiency. A condition of high white blood cell count, that's called leukocytosis, and that typically means an infection. High white blood cell count is typically in a few thousand more than the normal. So 15,000, 20,000 will be considered leukocytosis. On the other hand, a cancer of the blood called leukemia involves inordinate, very high amounts of red blood cells. So that could be something like, white blood cells, leukocytes. That could be something like 100,000 leukocytes, 500,000 leukocytes. Okay, not just a few thousand, but hundreds of thousands more than normal. Platelets are normally counted in hundreds of thousands, 250 to 400,000 per macroliter. A low platelet count is called thrombocytopenia. A high platelet count will be called thrombocytosis. Okay, and finally, we're going to briefly talk about a type of inherited anemia called sickle cell anemia. This is a mutation in the beta globin of hemoglobin, which results in a patient that cannot synthesize normal hemoglobin. This abnormal hemoglobin, under low oxygen concentrations found in tissues, changes shape into an elongated protein. Instead of round, is now elongated, which distorts red blood cells. Also, this abnormal hemoglobin cannot carry oxygen. So the cell now distorts its shape and turns into what's called a sickle shape. That's why the condition is called, sickle cell anemia. You can see here the sickle shape of the red blood cell versus the nice round shape it's supposed to have. So sickle cell anemia is an inherited form of anemia. The most common anemia, however, is due to a deficiency in iron. This is called iron deficiency anemia. This will lead to a reduced hemoglobin production. Pernicious anemia is due to a factor called low intrinsic factor that it's needed for B12 absorption. Hemorrhagic anemia is due to the loss of red blood cells. Hemolytic anemia is due to the destruction of red blood cells. So you should have an idea of these types of anemia and their causes. Von Willebrand's disease is a condition in which there's a lack of von Willebrand factor, which prevents the platelet plug formation of hemostasis. And then hemophilias are sex-linked traits in which the genes that code for the clotting factors are mutated and the patient has problems clotting blood. Because it's sex-linked, most of the individuals afflicted are males. So a female patient with a blood count of 3.2 million red blood cells per macroliter and a hematocrit of 32% is typically anemic because the red blood cell count is low. More likely the type of anemia may be due to a lack of iron, for example. If the hematocrit is 32%, that means that plasma is 68%. You should know what is the cause of sickle cell anemia and distinguish between leukocytosis versus leukemia. Okay. Due to blood incompatibility, sometimes a baby is born with jaundice, which is likely to deliver a baby with jaundice. An O-positive woman carrying a B-positive baby. An O-positive woman will have anti-B antibodies in their blood, so yeah, that could possibly result in a jaundice baby. An AB-positive woman carrying an O-positive baby should not harm the baby because the AB-positive woman does not make any antibodies. An A-negative woman carrying an A-positive baby, the The woman does not carry any specific antibodies against the A antigen. However, because she's Rh negative, being exposed to Rh positive blood would cause the production of anti-D antibodies that could potentially harm the baby. Yellow babies are put under UV light, jaundice babies, because UV light destroys bilirubin. Okay.